I just want to introduce our speaker for today. I'm really excited to welcome Dr. Mitchell Coleman here, who I just want to say received his PhD this past September. So congratulations. I've actually
It says candidate on there.
you have to change that. So I met Mitchell in 2023 and he came here to do some sampling for what is now his dissertation that he's going to be talking about today. Mitchell did his Bachelor of Science degree at Westmont College in Santa Barbara, and then his master's at Cal State University in Bakersfield. And then his PhD was done at the Evolution, Ecology, and Organismal Biology program at UC Riverside. Mitchell has actually done a lot of different things, and some of those things include being a lecturer at Cal State University in Bakersfield, and also an adjunct professor in biology at Bakersfield College. Mitchell has a long history with Tejon Ranch. Having done a lot of, having known it since his childhood, and became professionally affiliated with the Tejon Ranch Conservancy in 2015. This was through an environmental educational partnership impacting colleges and careers program, and this is what funded his master's research. And also permitted him to intern with the Tejon Ranch Conservancy. He later joined the Conservancy as a staff biologist and then served as the Conservation Science Manager and then on to Conservation Science Director. And now he has been appointed as of 2025, December, Executive Director of Tejon Ranch Conservancy. And so I want to welcome Dr. Coleman here to give a talk on using functional traits to predict ecological niche breadth and phylogeny in halophytic plants.
Thank you, Meredith. Really appreciate, thanks for inviting me. And I've really enjoyed getting the chance to speak to a lot of you today. Thanks for showing me around the garden. Really, the weather seemed to work out today. I started off with the heater on full blast with thick fog this morning, so thank you for nice weather. Mayor, I think, did you want me to start by reminding about the CalBot, or do you want to wait on that? Okay, who knows the California Botanical Society? It's not the garden, but the California Botanical Society. I'm the membership chair, and we're recruiting for council positions, including membership chair. It's answering emails. Slash putting videos on YouTube, slash doing whatever else needs to get done. If you are interested, please let me know. And I will train, I can do it, you can do it. And so I've been doing it for 5 years and I'm ready to hand the reins off to someone else. So if you're interested, please let me know. Okay, I did that. So, yeah, thanks for inviting me. It was my dissertation project, and I'll start with one other precursor. The society invited me to give this talk a few weeks ago on Zoom, and it had been 3 months since I cracked open the PowerPoint, and I realized, oh, there's some dust that settled on this thing, and I went too long. And so if I ramble, I apologize, but I think I'll do better this time. But always practice. My problem is brevity. I have a strong, it's not my strong suit. So I will promise not to ramble, but I probably will break that promise. Anyway, so let's advance this. Let's see, what button will advance? Oh, I probably need to just click it.
Might need to use clicker.
Oh, there it goes. Okay, so before I get started, I just want to acknowledge the many people and organizations that played a role throughout this process. My dissertation started in 2020, and what was happening in 2020, that was the pandemic. And so it was a really surreal time for a variety of reasons. And it was kind of a strange time to start a PhD. And so it was very isolating in a lot of ways, but I had a village around me nonetheless. And so if any of the people or organizations that are on this slide were not here, this entire project would not have been possible at all. And so I'm just super grateful. Committee members, my advisor, Luis Santiago, my colleagues at the Tejon Ranch Conservancy, my family, all mayor here at the garden, a lot of folks at various herbaria. If there was anything missing here, that I wouldn't be here. And so thank you so much for everyone. I'm really, really grateful. So in the context of global change, it's widely acknowledged that there has been and will continue to be variations in precipitation and temperature on a global scale. This map here on the right is one of the more recent global prognoses of patterns of plant mortality related to a broader trend of hotter and more frequent droughts over the last 60 years or so. So the purple points, thank goodness it's not washed out, it was during my dissertation. The purple points represent mortality events in the 1970s to 1980s, whereas the yellow to orange points represent massive mortality events from about the 1990s to about present. What you can see is that this trend is just exacerbating through time. It's getting worse. Now, of course, hotter and more frequent droughts are the broad two categorical variables, but that can have a wide range of cascading effects across all sorts of ecological scales, pollinator dynamics, you name it. All sorts of things can be influenced by changes in precipitation and temperature. Just one of those changes, and one that's not often talked about, is altered salinity patterns across the landscape. Already about 6% of the Earth's land surface is affected in some way, shape, or form by salt. This includes both natural landscapes as well as agricultural zones. And that's consequential, right? Because high soil salinity is lethal to most plants. It induces chlorosis, it inhibits water uptake, and it ultimately leads to mortality for most taxa. And of course, that has ecological implications. As areas become hotter and drier and conceivably more saline, what does that mean for how plants are able to physiologically persist in these conditions? It also has agricultural implications. In fact, a lot of the work that has been done in this wide category of global soil salinization has an agronomic context. And so a lot of agricultural areas are getting hotter and drier and more saline. So there's real food security and food production implications here as well. And so that gets us to halophytes. Halophytes are salt adapted plants. Now, they're often assumed to be these sort of ecological specialists, right? Because their overall strategy is to occupy these really salty conditions that most plants simply cannot tolerate. However, that categorization of specialization isn't quite true. Halophytes are actually quite diverse for a plethora of reasons. I'll just go through a few really simple ones. They are, let's see, there we go. They're functionally diverse. So they have different physiological mechanisms that enable them to persist in the context of high salt concentrations, from some halophytes on the very top, some halophytes that exude salt onto the surfaces of their leaves, to other halophytes that sequester salt in their vacuoles and cause their tissues to swell and become succulent. Still other halophytes sequester salt at the root level and largely inhibit salt uptake into the xylem altogether. So they're functionally diverse with different mechanisms. They're morphologically diverse with every major growth form, from little annuals to shrubs to full-size trees, even some lianas. They're phylogenetically diverse. So halophytes are about 0.4%, 0.5% of all flora. That's just over 1,200 species. But those 1,200 species are represented in over a quarter of described plant families. So halophytism itself is actually polyphyletic, meaning it has evolved multiple times across multiple lineages. However, there's a subset of 8 plant families where halophytism is extremely well represented. Those are those red families down below. So some halophytes are quite closely related, others are more widely diverged. They're geographically diverse, occurring across all major biomes and different environmental conditions, from extreme hot to extreme heat, all kinds of things, right? The only exception to that rule is extreme cold. They don't really occur in the polar regions. That's the one exception. Lastly, they seem to demonstrate quite significant ecological niche diversity, whereas some halophytes, yes, they really do seem to only occur in those extremely salty conditions. They depend on them. They do not occur outside of them. They're obligate. In other words, they have narrow ecological niche breadths, whereas other halophytes, they can occur in those salty conditions, but they don't necessarily depend on them. They're facultative. In other words, they have wider ecological niche breadths with regard to salinity tolerance. So for my dissertation, I was asking to what degree do halophytes specialize and generalize, and taking into account how some halophytes are quite closely related, others are more widely diverged. How does their phylogeny play a role in shaping those patterns of niche breadth? And ultimately, what are the potential responses to global change as areas become hotter and drier and more saline? So that gets us to chapter one. Chapter one was my field chapter, to salinity and beyond. I like puns. To salinity and beyond. This was a chapter where I got to spend, this was my pandemic chapter. I was largely outside doing a functional trait analysis, mostly in the San Joaquin Desert of California, along a salinity gradient. And so right here, I just want to do a quick primer on scales of study, okay? I was primarily concerned with two scales. The first scale is what I'll simply just call the functional strategy. When I say functional strategy, I just mean halophyte specialists versus halophyte generalists, okay? That's strategy. I was also interested in variation between populations within the same species, intraspecific trait variation. This was a really, it's kind of an emerging field in the world of plant physiological ecology. A lot of interesting things that you can look at there. I learned it's actually really hard to succinctly present intraspecific trait data. So I'm not going to talk about that, but I do have bonus slides. So if you have any questions about, I won't talk about that during the presentation. I will focus on the functional strategy level, but if you have any questions about that afterward, if there's time, please feel free to ask me. So for my hypothesis, I hypothesized that specialists and generalists had trait differences across scales, predicting that the functional traits of generalists would be more significantly variable among the different environmental conditions in which they occur, whereas the functional traits of specialists would not vary. They would be relatively labile across environmental conditions with regard to their functional traits. And I secondly predicted that the functional traits of specialists would be more tolerant of the absolute extremes, the absolute environmental extremes. And so to assess this hypothesis, I established 14 sites in the San Joaquin Desert of California. Here's a map on the right. These sites were generally more saline on the north end of the gradient and less saline on the south end. Also, mostly within the Central Valley, which is appropriately called the San Joaquin Desert. San Joaquin Desert, 12 sites were there, a couple of satellite sites, and that was just to achieve repetition for one species because it's not super common on the valley floor. Okay, so 14 sites, environmental gradient. I measured 15 species, 9 of which were halophytes and 6 of which were glycophytes or non-halophytes. I will get into that in a moment. And between 2021 and 2024, I did a variety of things. I, of course, measured environmental conditions, so things like soil salinity, soil pH, soil moisture, etc., as well as various functional traits to get at various axes of ecological strategy variation. So things like hydraulic traits, midday leaf water potential, turgor loss point, things like that, leaf economic traits, structural traits, all in relation to the environmental conditions. And I analyzed the data with a combination of stepwise multiple regression and ANOVA. So this is just a quick snapshot of the halophytes that were within the study array. Those that are outlined in red are what I call the specialist halophytes, and those that are outlined in blue are what I call the generalist halophytes. Now, I'll note, this was an a priori designation. It was a designation I made beforehand based on the environmental conditions in which these species occur. I grew up in Bakersfield, so I'm pretty familiar with where these species occur on the valley floor. You don't see the specialists outside of the absolute saltiest areas. You don't, they're obligates. Whereas the facultative, the specialists, they occur in the salty sites, but far more continuously across the gradient. So I justify that categorization based on those observations, as well as more broadly, several different community assessments that have been done through the decades. I also measured functional traits of these 6 co-occurring glycophytes. Okay, these are non-halophytic species, right? They cannot tolerate salty conditions, but I was interested in seeing how their functional traits varied outside of the explicit context of salinity. So they occur within the gradient, but they have different means of avoiding the problem of salt. Maybe they have avoidance mechanisms like deep roots, or they occur in little microsites of low salinity. So they occur in these sites. But I wanted to look at how their functional trait variation occurred, presumably outside of the environmental, the problem of salinity, sort of as an environmental control, if you will, okay? So for results, if you can't tell by now, everything is color coded, and this will be the same throughout everything. So the specialists are outlined in red, the generalists in blue, and the glycophytes in green. Okay, so specialists occurred in hotter and drier sites overall. So this panel here on the left is mean annual precipitation, and the panel on the right is mean annual temperature. What you can see is that specialist halophytes occurred in by far the driest conditions, generalists somewhere in the middle, and glycophytes way variable, but by far in the highest precipitation conditions. And the opposite trends for temperature. Specialists occurred in by far the hottest sites, whereas generalists and glycophytes actually did not significantly differ, but you can see a huge gradient for the glycophytes. Now, this was not a surprise, but specialists also occurred in saltier and more alkaline soils. This is mean soil salinity here on the left panel and soil pH on the right panel. So specialists occurred in saltier conditions relative to generalists and glycophytes. Interestingly, oh, I forgot to note, these letters here above the bar charts are post hoc differences between the groups. So if the letters are different, that means they're statistically different. I should have explained that. One of those things you forget. Okay, so yes, specialists occur in the saltiest sites, and generalists and glycophytes actually did not significantly differ, but you can see pretty significant range for the generalists. Now, for soil pH or alkalinity, specialists occurred in by far the most alkaline conditions, and generalists and specialists did not significantly differ. Okay, so this is our first functional trait. This is midday leaf water potential. Leaf water potential is essentially the water status of leaves at the particular time of measuring. In this case, midday during the fall of 2021. Who lived in California during the fall of 2021? Someone mentioned at lunch today, I think it was Naomi, mentioned she thinks about years in terms of the weather. That was a rough year. It was really hot, really dry. I think there were, brownouts, the state electrical grid was suffering. These were the hottest conditions and driest conditions in which these texts, it was a record breaker as far, at least in my understanding. So basically there it is. It's jumping ahead, Mayor. So the lower down on the y-axis, the less hydrated the leaves are. Okay, that makes sense? Okay, so specialist halophytes had by far the least hydrated leaves, whereas generalists were somewhere in the middle, and glycophytes were by far the most hydrated. Okay, and this can be influenced by a wide variety of environmental conditions. Okay, it's kind of an interesting trait. Okay, specialist most dehydrated. Okay, now this is the same data, midday leaf water potential as a function of soil salinity. So soil salinity is increasing there to the right of the figure. So generalists were the only functional group that had a relationship. In other words, as soil salinity increased, their midday leaf water potentials decreased. They became less hydrated at higher salinities, whereas specialists were stable across conditions. Okay, so as soil salinity increased, their midday leaf water potentials did not vary. And I won't talk about glycophytes because that's a harder question to answer. And it skipped ahead, but I'm ready for it. This is soil pH. Okay. Oh, another thing I forgot to mention. Look at those R-squared values. Doesn't explain very much, does it? So this was actually surprising to me. I thought that soil salinity would explain much more of the variation than it actually did. There were significant associations, but it didn't explain all that much of the associations. A lot of the variance was not explained by soil salinity. That's why I put this in here. So this is soil pH as a function of soil salinity, or no, so midday leaf water potential as a function of soil pH. So same trend. Specialist halophytes were similarly stable across conditions when it came to soil alkalinity, but look at the generalists. They actually were more hydrated with increasing alkalinity, but look at the rest, close to 30% of the variation was explained this time. So a lot more of the variation was explained by pH than temperature. This was a surprise to me. Perhaps I was biased, but I thought that, so these are halophytes, a lot of their variation would be explained by salt. Not the case, ostensibly not the case. Now, this is the modulus of elasticity, which is a fancy way of saying the rigidity of the cell walls. So classic plant physiology tells us that plants with more rigid cell walls will be more resilient to environmental extremes. And it turns out that specialist halophytes have, skip it ahead, specialist halophytes had the thickest cell walls, the most rigid cell walls, whereas generalists and glycophytes did not significantly differ from one another. Now, somewhat similar to midday leaf water potential, generalists halophytes had less rigid cell walls at higher salinities, but still not very much of the variance was explained by salinity. Take a look at those large squared values, but also specialists were stable across conditions. But if we take a look at the association between cell wall rigidity and mean annual precipitation, we can see a relatively higher proportion of the variance explained, at least for generalists, okay? These are the standout trends, by the way. And finally, this is leaf turgor loss point. Leaf turgor loss point, this isn't quite true, but it's a handy way of thinking about it. Leaf turgor loss point is the point at which your houseplants would be dead if you forgot to water them. This is the point at which they would wilt and long since have died. Okay, so interestingly, between specialist and generalist halophytes, there was not a significant difference in leaf turgor loss point. Significantly lower than glycophytes, but not significantly different from one another. When we look at the association between turgor loss point and salinity, we see some interesting trends. It was actually the, nothing varied with turgor loss point and soil salinity. No trends there to speak of. There was more something to say for soil pH. Generalists were more variable when it came to turgor loss point and soil pH. But of all these trends, these are just data to think about. Of everything I've shared with you, I would like you to pay closest attention to this. This is, I think, the key takeaway. This is midday leaf water potential on the left and turgor loss point on the right. These two functional traits have the same units, so we can plot them and analyze them together. Specialist hail, are those coyotes?
Yeah, it is.
Love that. So specialist, I'm gonna be thinking about coyotes now. So specialist halophytes were the only functional strategy that had midday leaf water potentials beneath their turgor loss points. I'll say that one more time. Specialist halophytes were the only functional strategy that had midday leaf water potentials beneath their turgor loss points. What's going on there? If you think about that for a second, turgor loss point is, you can think of that as the point at which plants wilt, okay? This was the fall of 2021. These plants are past turgor. They're not only alive, but they were thriving past the point at which they have ostensibly lost turgor. This is unusual. It has been documented in the literature before. A lot of our chaparral species, I saw you had some Ceanothus cuneatus here in the garden. This is, Ceanothus cuneatus is famous for this. I think Andinostoma can do this too. Some plants, chaparral, I think is a good example. A lot of chaparral species have this ability to survive past turgor, which just blows my mind. Specialist halophytes ostensibly have this ability as well. Okay, generalists and glycophytes, we're not demonstrating those patterns. Okay, so for conclusions, specialists, I'm pretty comfortable saying, occurred in higher environmental extremes, saltier soils, higher temperatures, lower precipitations across the board. Specialists who occurred in more environmental extremes. The specialist traits often reflected those extremes, okay? They had the lowest midday leaf water potentials, the more rigid cell walls, the functional traits were tied in many ways to the environmental extremes. Generalist halophytes, meanwhile, were more variable across conditions with regard to their functional traits, whereas specialists were more stable across conditions. And this, by the way, actually, I'll wait for a second to say that. I'll go ahead and say it now. Stable across scales. I know this is only talking about the strategy level, but this is a phenomenon that transcends scales. Salinity, this was hard for me to admit. You can't, I don't want to say this, but salinity was not the most important driver. It was actually, if there was one environmental variable that stood out, that really had a large influence, a disproportionate influence on functional traits of halophytes, it was alkalinity, soil pH, not soil salinity. This is interesting. And there's more that could be said there, but in the interest of time, I won't. Finally, key takeaway point again, specialists ostensibly had this ability to survive past the point at which their leaves have lost sugar, which in the context of global change, environmental extremes, that's really something to think about and something that could be looked into further. So in the end, I conclude that my hypothesis was supported. Functional traits of generalists more variable across conditions. Functional traits of specialists were more stable across conditions, and again, specialists were more tolerant of the extremes. So for chapter two, this, if what I just told you was bottom up, this is top down, okay? This was an opportunity to really zoom out and look at the world. I had never done this before. It was very, very educational. So I had the opportunity to examine halophyte functional trait variation in the context of phylogeny now on a global scale. So this was fun and a real challenge. A quick reminder, halophytes are 0.4% of flora. That's just over 1,200 species, but those 1,200 species are represented in over a quarter of described plant families. So halophytism is polyphyletic, but there's some halophyte families, these red ones, where halophytes are extremely well represented. I can't stop thinking about coyotes now, I'm sorry, that's just so cool. Okay, but halophytes are really well, there's a subset of halophyte plant families that are extremely well represented. Okay, so this was the challenge. I know a lot of you, you're my mentors for this. I was not trained I took basic plant taxonomy, I understood the core concepts of phylogeny, but this was not my training. One of my advisors impressed upon me that when you're doing an interspecies analysis of any note, one that holds its salt, pun intended, you have to account for phylogeny, okay? Because general statistics, and I know I'm preaching to the choir here, but conventional statistical models assume equality of variance between different groups, but that's not necessarily the case, is it? So you have to use statistical models that account for phylogeny. This was hard. I wasn't trained how to do it, but I learned. So I did this. You have to account for phylogeny. This was one of the primary goals of this chapter. So my hypothesis was that halophyte trait expression is influenced by both niche breadth, in other words, strategy, as well as phylogeny. And I predicted that specialist halophytes would be more phylogenetically structured relative to generalists. I also predicted, much like chapter one, that number one, specialists would exhibit higher tolerance of environmental extremes and also show weaker trait environment associations or be more stable across conditions after accounting for phylogeny. So to assess this hypothesis, I used the eHalof database. The eHalof database is one of my best friends. It's administered out of the University of Sussex by Dr. Tim Flowers, who's one of the leading global experts on halophytes. It was up until the early 2000s only a paper database, but they made it available online in the early 2000s. It's been around for a little over 20 years now. Again, administered out of the University of Sussex. You can go on there now and you can contribute. If you have any halophytic plant data, feel free to contribute. So you can access this database, which is what I did. When I accessed this back in 2024, there within the database were 96 families and just shy of 1,200 species, which is basically halophytes, right? 1,200 species, it's 99% of the known halophytes. It's quite a complete data set. There were, of course, gaps in the data, but it's pretty good, Each species in the database is essentially an N of one, so it gives you broad metadata with literature tied to each point, okay? So variables associated with each species included things like growth form and habitat type, photosynthetic pathway, and really importantly, maximum salinity tolerance. That maximum salinity tolerance, it's a continuous data point, mind you, for each species. That became really important. because I had never done a meta-analysis before. I don't think anyone has ever gone over the whole world for their field site. That would just, well, that'd be crazy. No. So I had to have, unlike chapter 1, I had to take a different approach when it came to characterizing halophyte strategy. And the way I did that was a proxy method by using the maximum salinity tolerance value that was present within eHalo. So species within the database that had maximum salinity tolerance values that were greater than 8 millisiemens per centimeter. This was kind of on the upper end of what I personally documented in chapter 1. So species with maximum salinity tolerance greater than 8 millisiemens per centimeter, I categorized as specialist halophytes, whereas species with less than 8 millisiemens per centimeter, I characterized as generalists. Now, this is a proxy. It's not perfect. There's actually quite a few holes in it. You're probably already thinking, okay? So we will acknowledge that, but we will also acknowledge that it is kind of supported by the literature. I did a fairly exhaustive literature review. Folks have been studying halophytes for decades, way before I came along. All kinds of different interesting research, largely in an agronomic context, really interestingly, but different groups of halophytes, hydrohalophytes, obligate halophytes, facultative halophytes. There's a lot of continuous data out there. Okay, this 8 millisiemens per centimeter cutoff that I used is about the middle ground. Okay, it's a moderate approach. It's not the extreme end on either way, so I justify it on the basis of that. Okay, but again, it is a proxy. So then I use who's used the tri-plant trait database before? Nobody. Well, it's also my best friend. It is a global repository for functional traits. You can upload your data yourself if you want to. Millions upon millions of different functional traits are available within this database. So I queried the tri-plant trait database for these specific 817 species, which traits were the most represented for these 817 species within the database. Turned out there were 5 that were quite well represented. Leaf dry matter content, which is mass of dried leaves, specific leaf area, I'll talk about that in a second. Leaf width, leaf traits, seed mass, and plant height. Those were 5 that I ended up using because they were so well represented. They're simple traits, but quite robust data, okay? There was also pretty good environmental data available in the plant trait database as well, meaning precipitation, temperature, elevation, etc. Okay, so got functional trait data with the tri-plant database, and finally I tied in that phylogenetic component using phylogenetic independent contrasts. Okay, these are Brownian motion models that, to be honest with you, I don't know how they work, but I trust that R does. And so I had to learn how to. You probably, do you know what I'm talking about? I think you know more than I do about how these models work. I used them, and this was the way that I used for accounting for phylogeny. Okay, so for results, again, generalist halophytes are in blue, and specialist halophytes are in red. So specialist halophytes were overwhelmingly phylogenetically structured. Over 80% of the specialists of the global halophytes were specialists. Okay, now there's a lot of things that could be said here. It could again have something to do with the way I treated halophyte strategy, but at least making this assumption, most halophytes fall within the specialist strategy. More things that could be said also, probably half of the plant families were only specialists. Not the distal tips, by the way, are families. I know it's probably a little bit difficult to see, but that's distal tips on the far end. Half of the plant families, at least the species within the database, all of them were specialists, okay? Fully half. The most species-rich families were also dominated by specialists. These were not families that every single species was a specialist, but they were dominated by specialists. So these are like the Myrtaceae, the Fabaceae, the Poaceae. These families were dominated by specialist halophytes. And lastly, but not least, probably a bit difficult to see there, but the most species-rich halophyte family, does anyone want to guess? Amaranthaceae, which is my absolute favorite family in the world. So you'll hear about that a little bit later, but 20% of global halophytes were amaranths, which was really interesting. Not a huge surprise, but that's just because they're really awesome. Okay, so specialists were more likely to have the C4 photosynthetic pathway. If you need a little bit of a refresher on C3 versus C4, I'll worry not. I will give you a refresher in the next chapter. But suffice it to say that the C4 photosynthetic pathway is adapted to more extreme environmental conditions, hotter, drier, etc. So specialists, after accounting for phylogeny, were more likely to have the C4 photosynthetic pathway. It was marginal, but more likely. Again, everything I'm showing you is after accounting for phylogeny. Specialists also tended to have the shrubby growth form, whereas generalists tended to be more annuals, okay? Thinking about this phylogenetically, you could make definitely C4 plants evolve from C3 plants. That's pretty clear. You could also think about shrubbiness coming from annuals. There's lots that could be said here, but specialists tended to have the shrubby growth form relative to generalists. There is a lot to look at right here. I know it's not good practice to put multiple slides on one figure, but I'll make it really simple for you because strategies did not differ with regard to any of the environmental conditions or any of the functional traits. The top row are environmental conditions, the bottom row are functional traits. The only exception to that was maximum salinity tolerance, but that tells us nothing because I'm the one who imposed that difference to begin with. So can't infer anything on that basis, but with regard to all the other environmental conditions, with regard to all the functional traits that were analyzed after accounting for phylogeny, there were no significant differences between the groups. This was interesting, hard for, again, I didn't want this, but this is what the data said. But in terms of environmental associations, I'm gonna go quick here because there's a lot to say. So this is the next series of slides. All the solid lines denote a significant correlation. The dashed lines are insignificant, okay? So this, which one is this? This is leaf dry matter contents, the mass of dried leaves for every environmental variable. Okay, so generalists and specialists had similar responses for leaf dry matter content across all of the environmental conditions. Okay, they did not significantly differ relative to one another. Generalists, meanwhile, had a stronger response with specific leaf area. Specific leaf area responded more strongly to environmental conditions relative to generalists. I'm intentionally moving quick with these slides. Specialists also, the leaf width in specialists, meanwhile, I think it was the only one. There were none for the generalists that has significant response. Specialists had a stronger response to the environment with regard to leaf width. Likewise, specialists also had a stronger response when it came to plant height. I don't think there were any generalists. Maximum salinity tolerance was the only exception. So specialists had a stronger response when it came to plant height. And finally, seed mass. Specialists were the only, I think it was mean annual precipitation. So the mass of specialist plants, the seed mass of specialist plants were lower at higher precipitation. That was the only one after accounting for phylogeny. I know that was a lot. And I went through that quickly for a reason, and I'll explain why. So, in conclusion, specialists were more closely related overall. This could have been biased, I will admit, by the way I treated halophyte strategy. But again, making that assumption, specialists were more closely related, much more phylogenetic structuring. There was a broad association between halophyte specialization and the C4 photosynthetic pathway and the shrubby growth form. In the context of environmental extreme tolerance, this is indicative, after accounting for phylogeny, of specialists being able to tolerate more extreme conditions. This is only categorical variables, but the continuous analyses did not affirm that at all. It was there were no significant differences between the groups. When it came to trait environment responses, it was really more mixed. I think there were 12 for specialists. There were 12 significant trait environment associations, only 4 for generalists. So I wanted to say that there was a stronger association for specialists, but my committee said no. And so it's really more, it's more of a mixed, it's mixed. There's really it's hard to pick out a trend that leaps off right here. So I end with saying that the hypothesis was partially supported. So specialists, at least with regard to how I treated it, exhibited more phylogenetic structuring in the context of the categorical analyses with specialist halophytes being tied to C4 photosynthesis. That's again indicative, possibly, of more environmental tolerance to environmental extremes, but the continuous data again did not bear that out, and there was really not much of a pattern when it came to trait environment responses association. So for future studies, this is the one that I'm going to wait the longest on to begin to publish, because it's if you can't tell, this one was my weak link. It's difficult. I learned a lot I think I learned a lot. There's a lot that could be said here. In the future, I think that we will get away from halophyte strategy and just run a through line through all the data. Another thing I'd like to do is to try to use different cutoff points. I used 8 millisiemens per centimeter here, but maybe one lower, 4, maybe one higher, like 12, and seeing how that shapes the responses for the data. Okay, so there's a lot more that could be done here, but still, it was a very useful exercise, and I learned how to make a phylogeny on R, so that was fun. Okay, so for chapter 3, this is my favorite. This is my absolute favorite, and I love this image because it shows the break in the clouds. We're getting near the end here. I get to talk to you about my absolute favorite genus in the world, which is the genus Atriplex. Okay, so quick primer on the genus Atriplex. These are the salt bushes. They are the most diverse halophytic genus. Okay, so there are about 260 species, and they really mirror halophytes in a lot of ways. They occur across all kinds of environmental conditions across the globe, really all over the world. There's one that's, I think this is Denmark, way up here. I would like to go there one day, but they occur all over the world in all kinds of environmental conditions. There are specialist salt bushes and there are generalist salt bushes, okay? Their phylogeny has long perplexed taxonomists for a variety of reasons. Take a look at the pictures here. Would you say that these are all pictures of the same species or different species? Hard question. All the same? All the same? We're in consensus, all the same. You're right. I couldn't stump you, okay? In fact, these are all taken from the same population on the same day, no color editing. A huge amount of morphological variation in the leaves. Okay, a lot of variation. If you think the flowers will help you if you like to keep stuff out, no. The Jepson's not. The Jepson manual, dichotomous key will not help you. The flowers are, I'm sure you know, these beautiful apetalous bulbs on the end of the inflorescences. And you get to a point where a dichotomous key is not going to be much more use. You have to go molecular, which is where my skill set is the ends. Okay. So what would you say for these? Would you say that's all the same species or different species? They're all different. They're all different, but they look kind of similar, right? I think they're beautiful, but people make fun of me for thinking they're beautiful. Does anyone agree with me that they're beautiful? I thought that well, higher proportion of people said yes than probably ever. So they also have a really complex degree of polyploidy and hybridization and floromorphogenesis. What else can be said here? If you use molecular approaches, you can still run into problems. As a result of all these things, the genus has undergone quite significant revision in the last 100 years, from over 400 species in one of the earlier treatments by Holland Clements in the 1920s to about 260 species today. I say about because it's still an active area of research. I actually got to travel to Germany and meet Gudrun Keiderreich for this project. She's the global saltbush expert. That was cool. But about 260 species. Her lab group is one of the leading groups working on saltbushes and still an active area of research. One of Gudrun Keiderreich's postdocs at the time, a guy named Anze Zerdoner, this was his work. Okay, so there's an interesting story to be said about how saltbushes radiated in response, how they evolved. They're one of those unique groups that are majority C4 photosynthesis. This is interesting. There's emerging resolution now that salt bushes emerged from Central Asia 10 to 15 million years ago in response to acidification and higher temperatures that were going on throughout that epoch. Okay, by evolving C4 photosynthesis sequentially. There's emerging clarity about how salt bushes do that. Still a lot of interesting questions that could be asked, but I also got to meet Hansi, very cool guy. So I promised a quick primer on C3 versus C4 photosynthesis. I don't assume that you don't know this, but if you need a refresher, here it is. Okay, C3 photosynthesis is by far the most common photosynthesis type. 95% of plants, these are plants that do best in more mesic, cooler conditions. They accomplish photosynthesis largely in the mesophyll cells using a very cool enzyme called rubisco. I know this is a review, but rubisco. C4 photosynthesis, on the other hand, is an extreme minority of plants, about 3%. These are plants that are adapted to high light, high temperature, high drought conditions. They also do photosynthesis with rubisco, but they achieve it somewhat differently. They undergo it additionally in the bundle sheath cells, and they use an additional enzyme in the process called PEP carboxylase. PEP carboxylase is important. C3 plants don't have it. That's important because PEP carboxylase discriminates against the atmospheric isotope of carbon differently than rubisco does. As a result, if you measure the carbon isotopic composition of C4 plants relative to C3 plants, you get different distributions. And this has been an emerging field over the past 50 years or so, stable isotope ecology. If you measure the carbon and it's not the only application here, but if you measure the carbon isotopic composition with a mass spectrometer in the leaf tissues of C3 plants versus C4 plants, you should, and I'll precurse that, you should get different distributions because of PEP carboxylates discriminating against the isotope of carbon differently, but you should. One of the interesting things about this project, early on, I took samples from the UC Riverside herbarium, and there were a handful of samples that did not, that essentially fell between the established distributions of C3 versus C4. I call this etra-perplexing. Etra-perplexing. What's going on here? I was taught that you have simply C3 plants, C4 plants. They should be entirely separate from one another. What is going on? I admit I got unreasonably obsessed with this. It became very, very interesting. So it turns out that thinking about C3 versus C4 categorically in a binary is not necessarily the right approach to take. C4 plants obviously emerged from C3 plants, but it didn't happen overnight. It represented selection pressure over the course of millennia, lots of different transitional states in between ancestral C3 and derived C4. Okay, so thinking about that in a binary sense is not necessarily right. And some plants are still going through this process. Some plants are still evolving C4. Okay, one of the sort of model systems, model genera is the genus Flavaria, which is in the sunflower family, which is well known as having quite a number of C3, C4 species. Okay, so this again, wow, I think this is really cool, really cool. So for my hypothesis, I hypothesize that photosynthetic pathways and atroplex are related to niche breath. And I predicted, number one, that C3 salt bushes would be associated with halophyte generalization, whereas C4 salt bushes would be associated with halophyte specialization. Okay, does that make sense? Okay, secondly, I predicted that C3 generalist salt bushes would be more variable in response to the environment relative to C4 specialist salt bushes. And finally, I predicted that any intermediate salt bushes, species that had carbon isotopic values in the middle range, would demonstrate intermediate or middle range responses between those two. Okay. So for methods, this was my favorite part of my dissertation. I used carbon isotopes, obviously, to determine photosynthetic pathway. I collected well over 1,000 samples, but ultimately only ended up analyzing 385 This is where I get to thank Mayor. So Mayor let me in here at the herbarium, got to see her for a day. It was my first time in the garden. Got to go to Jepson. I even got to go to Germany, Munich, Germany, and spend 2 weeks there. I have a fun story to tell, but I won't just right now. Maybe ask me later. But this was my first international science trip, which was so much fun. Very, very impactful for me personally. Okay. So ended up measuring 385 samples, 172 species. So there was repetition. It ended up being about 66%, 2/3 of the genus, about. So representative. Okay. I then incorporated pathway determination with number one, Atroplex niche breath or halophyte strategy with environmental data. So I determined halophyte strategy somewhat more similarly this time to the way I did it with chapter one. These were using herbariums. I imagine most of you have worked in herbarium before. One of the handy things about herbaria is that they tell you where they came from, usually. People who don't, I'm like, come on, why didn't you write down where you collected this? Usually they tell you where they collected the specimen from, right? So you can tie that specimen to a discrete environmental condition. Usually, sometimes they even say right on there, occurred in salt playa. Thank you. I can now call you a specialist. Okay, so based on, it's kind of an interesting game, but ended up being able to tie halophyte strategy to each sample that I ran based on where it was collected from. Okay. And I also tied in environmental data with WorldClim, which is a global algorithmically derived data set of weather conditions, climatic conditions. Really fun to use that. I got used for that for environmental data, and then I used the ISRIC soil database, which is a global repository for edaphic data. Okay, so I tied in environmental and edaphic data with those two data sets respectively. So for results, C3 blue, intermediate yellow, C4 red. So this is the global photosynthetic pathway of Atroplex. Perhaps unsurprisingly, over 80% of global salt bushes fell within the C4 pathway. This is not new information. This was known. About 15%, 16%. The published value is 17%. So this is pretty close to published values. Those two things not new. What was new? 5%. 5% were intermediate. I will add that this has not been done before on a global scale for salt bushes using a mass spectrometer. A lot of the treatments were done with a good old-fashioned hand lens looking for Kranz anatomy. And that doesn't necessarily work, right? So this was the first time, at least as far as I know, that it was done globally for salt bushes using this particular method. So we got about 5% that fell in between. One, I have to admit, you know how advisor, they point out your weak points, and my committee was very good about that. So if you look really quickly at the histogram here, you'll see that the tail end of the intermediate distribution is actually tied to C4. And my advisor wanted me to fold it over into C4. He had good reason for that, but I begged and pleaded for the purpose of the dissertation to have permission to just continue to count them as intermediate, because this was like 5 samples left that were, you might call true intermediate. It wasn't enough to do an analysis. So I really, really wanted, I was obsessed. I had an agenda. I really, really wanted to look at this. And so I got permission. My committee was okay with it, but I will mention that take the intermediate analyses with a grain of salt bush, okay, because of this. Okay. So this is just a quick map of the global halophytes that I ended up collecting. As you can see, 2/3 of the genus, but again, representative, all the major groupings of salt bushes on a global scale. And you can pick out some interesting things just by looking at the map. For example, a lot of the C3s in blue, if you can see those, you don't even see my mouse. A lot of the C3s and blues are north of 20 degrees or south of 20 degrees. Okay, and they're often tied to coastal regions as well. Okay, the interesting things that you could pick out there, same data, but with a mean annual temperature layer applied over it. You can't really pick out a lot of the trends unless you zoom in, but I still think it's pretty, so I put it in there. And then same thing with mean annual precipitation. There's some really interesting patterns that you can pick out there. I just think these maps are pretty, so I like maps. Okay. So C4 and intermediate salt bushes were associated with saline habitat, just in terms of the categorical habitat type, where these are variant accessions were collected from. The gray portions of these stacked bar charts represent saline habitat. And so intermediate and C4 salt bushes were overwhelmingly associated with saline habitat, whereas C3 salt bushes really occurred across a wider breadth of conditions, as you can see. Okay, this was, again, categorical. C4 intermediate salt bushes also were overwhelmingly much stronger trends than the previous analysis I did for chapter 2. C4 and intermediate salt bushes were overwhelmingly halophyte specialists, whereas C3 salt bushes were overwhelmingly halophyte generalists, categorically. So this is continuous soil salinity as a function. This is soil salinity here on the y-axis. Okay, so interestingly, this was deja vu for chapter 1. Soil salinity continuously in continuous data did not vary by pathway. Okay, they did not significantly differ, but if you look at the C4 pathway, you can see that there are some pretty extreme outliers. I will also note that a global salinity data set, a really good one, doesn't really exist. If you know of a global soil salinity data set, please let me know. This is the best one I could find. I suggest that there's more going on here than what this one analysis indicates, okay? But at least how I analyzed it with the available data, soil salinity did not vary by pathway. In terms of responses of carbon isotope 13 to salinity, so this is carbon isotope here on the y-axis, so higher up is skewing C4, lower down is skewing C3, soil salinity increasing along the x-axis. So pathways, with regard to their carbon isotope 13, did not vary across salinity conditions. Okay, none of the three pathways. Similar trend here. Well, no, different trend. C4 and intermediate salt bushes occurred in more alkaline soil. This was actually similar. If you remember, this was kind of similar to chapter 1, right? So C4 largely specialist salt bushes were associated with higher alkaline soils relative to generalists, and intermediate salt bushes were very closely tied to C4. C3 salt bushes varied across alkaline conditions. And so as soil pH increased, there was a very marginal uptick in carbon 13 in C3 salt bushes. They skewed a little bit more C4 at higher alkalinities. Not enough to become C4, but interesting, right? It's indicative. Okay. C4 and intermediate salt bushes also occurred in drier habitats. Are you picking out there's some coupling going on between intermediate and C4? Salt bush, remember, grain of salt bush. There's that coupling. Intermediate and C4 salt bushes also occurred in drier habitats. C3 generalist salt bushes occurred in higher precipitations. C3 salt bushes varied across conditions, intermediate and C4 did not. So as mean where precipitation increased, C3 salt bushes skewed more C3 along the grade. C4 and intermediate salt bushes occurred in hotter habitats as well. Okay, so this is mean annual temperature with C3 salt bushes occurring in moderately cooler habitats. But pathway did not vary across any of the temperature conditions. I know I'm moving quick here, but here's the interesting one. C3 and intermediate salt bushes, this was the only exception. Across all the different environmental variables that I measured, the only one in which C3 and intermediate were the same and different from C4 was in maximum temperature, the absolute hottest that these sites get. Okay. Which is interesting. Maximum temperature seems to be playing a role. Again, that's the only one. Pathways did not vary in response to the hottest, the maximum temperature, the hottest that these sites get. So for conclusion, there was an association between C4 photosynthesis and halophyte strategy. So C4 and intermediate salt bushes were associated with both salty habitats and halophyte specialization, whereas C3 salt bushes were associated with less salty habitat and more halophyte generalization. The pathways also partitioned across the environments. There was basically a consistent trend across all the data for C4 and intermediate salt bushes to be very similar every time, except for maximum salinity tolerance. Maximum temperature was the one exception where C3 and intermediates were not significantly different. C3 salt bushes were also more variable with regard to their carbon isotope 13, C4 specialist salt bushes were more stable across conditions. Very much like chapter 1, salinity was not the most important driver. Again, it was alkalinity. Soil pH drove a lot more of the variation relative to salinity. So salinity is not necessarily the most important factor. But overall, I suggest that my hypotheses were supported. Photosynthetic pathways and a related niche breath. C3 salt bushes are associated with generalization. C4 salt bushes associated with specialization. C3s more variable across conditions. C4s specialists more stable across conditions. The intermediate, the prediction of intermediate salt bushes having intermediate ranges that didn't happen. Okay. So for fun, I probably over time, but final conclusions. Number one, specialist halophytes were more tolerant of the environmental extremes, whether it was soil salinity measured in C2, maximum temperature here in California, whether it was on a global scale, specialist halophytes associated with the C4 photosynthetic pathway for salt bushes, C4 specialist salt bushes occurring in hotter and drier conditions. Specialist halophytes were more tolerant across environmental extremes across scales. Generalist traits were generally, I'm using general twice there, but generalist traits were generally more variable across conditions, whereas specialists were more stable across conditions. Okay. Photosynthetic pathways were strongly influenced by halophyte strategy. So for both chapter 2 and chapter 3, there was an association between C4 photosynthesis specialization, C3 photosynthesis generalization. Phylogeny plays a large role in structuring halophyte traits, but this was again the weak point. I don't think my proxy holds its salt, proxy approach of halophyte strategy. There's a lot more that could be done here. Okay, but phylogeny does play a role in structuring these patterns that we're seeing on a global scale. And finally, I'll leave with parting thought. Specialists and generalists seem to show contrasting responses and will continue conceivably to show contrasting responses and resilience to global change. There's one more picture I want to show you. This was one of my field sites. It's in the San Joaquin Desert. It's called the Semi-Tropic Ecological Reserve. It's actually a retired pistachio orchard that they let die back in the 70s. Okay, they took out all the wood. Well, they let it die because it got too salty. It got too salty and it was killing the pistachio, so they retired it. And if you can kind of look closely, you can see the old hedgerows there. Okay, you can see how salty it is. This was my saltiest field site, actually. What has colonized or succeeded that pistachio orchard entirely specialist halophytes. That's not necessarily a scary thing, but this is a trend that could become more common in the context of global change. It's not entirely unlikely for areas, especially low-lying areas, to have exponentially higher salinity. There's some interesting habitat here. In fact, just a few weeks ago, there was an endemic alkaline saline scorpion that was described near here. There's fairy shrimp that occur in intensely briny water that also at this site. So these systems are habitat. They're interesting habitat. They have a role to play, but they could become more common. So what is interesting to think about all of this in relationship to halophyte strategy. So with that, I will end, and I probably went over time, but if there's any questions, happy to answer them.
So let us thank Dr. Coleman again for his wonderful talk.